Okay, guys, time to start. You guys ready for acid base balance? Okay, couple of very important topics that we will a couple of very important topics that we will see happen again in our acid base lab, and let's just go over them and refresh really quickly. So we have agreed in the past that our renal system, comprised of the kidney parenchyma being the main cells of the renal system, and then we have accessory organs like the ureters that will pass this urine fluid that has formed, and the tubules are made up of epithelial cells, and then they pass this urine to be stored in the bladder and then properly voided with stimulation from your cerebral cortex that will override the distension signals that have been sent from your bladder, distended with so much urine fluid compressing against the boundaries and the circumference of your bladder, and that will feed into your spinal cord integration neurons, who will send an output to cause the immediate voiding and release of the urine. But all of that will be over ride by your cerebral cortex to properly find the proper time and place for you to void that urine. Okay, so now let's go back into this nephron. We are dealing a lot with the nephron. We have mentioned how the nephron is a site where there will be the formation of two fluids, and that takes place with two separate capillaries. So we have this very important structure known as the renal corpuscle. Why is the renal corpuscle such a special structure? Because it is made up of two different structures. What are the two different structures that make up our renal corpuscle? What are the two structures that make up our renal corpuscle? We have a tubular structure and a vascular structure. So a vascular structure in red, these are blood vessels. So you have capillaries. So you have the capillaries that are fenestrated, these capillaries that are fenestrated, and behind the capillaries, you have a basement membrane that is rich with podocytes, that is rich with proteoglycans, and behind the proteoglycans of your basement membrane, you have food processes known as the podocytes. And this makes a three-layer barrier. The first barrier is our epithelial capillaries, these holes, these fenestrations. Then we have our basement membrane made up of proteoglycans. And the last stepwise filtration barrier is your podocytes. And we said that these three layers do not only form mechanical filtration barrier, they also form a chemical barrier. Can somebody explain that to me? How? How are these three layers in your glomeruli, the capillaries, the basement membrane, and the podocytes, form two different kinds of separation? They form a physical, mechanical separation and a chemical separation. So how do they perform that? How are these three layers forming physical and chemical barrier? Physical and chemical barrier. Explain that. Short answer. Explain how are the three layers, the capillaries, the fenestrations, and the basement membrane, the capillaries, the basement membrane, and the food processes of the podocytes. Those three represent for me physical and chemical barriers. Explain. Or take a shot. Yes. Excellent. So the three of them do that. Very good. So, physical barrier, they physically, mechanically block the passage of albumin proteins. They pass it, that they block the passage of red blood cells. So they are very important, and not just the capillaries. It's the capillaries, the basement membrane, and the food processes, the podocytes. So that's physically speaking. How about chemically? How do they provide a chemical barrier? They provide a chemical barrier through charges. So the endothelium has a lot of amino acids that are what charge? Negative charge or positive charge? Negative charges. The proteoglycan that is found in your basement membrane is made up of negative charges. And the food processes, the podocytes, they are also enriched with negative charges. What charge is are the plasma proteins and the red blood cells and the lipoproteins that are carrying inside their core are lipids, the lipoproteins. They are all positively charged. So it makes perfect sense that these three layers, they physically block them because they are too small for their passage, and they also chemically repel them. So I'm sorry, these are negative charges. These are negative charges. The plasma proteins are also negative charges. The lipoproteins are negative charges. Red blood cells are negatively charged, and the lipoproteins are negatively charged. So same charges repel. So the glomeruli will repel the red blood cells, and also the food processes will repel, and also the basement membrane, because it's rich with negatively charged proteoglycans, will also repel these subjects. That's why if you see them leak into the urine, if you see them, this is feeding into this is my bladder. So this is fluid number one. That's urine. Fluid one. That's urine. And that's fluid two. That's blood going to the renal vein and going to the right heart. So if you see plasma in the urine, in the bladder, if you see red blood cells in the urine, in the bladder, if you see lipoproteins in the urine, in the bladder, that means they have defied the physical barrier of not one barrier, but three barriers. They have defied the chemical electrostatic repulsion provided by your glomerular capillary endothelial cells and your basement membrane made up of the proteoglycans and your food processes and podocytes. They have defied three physical barriers and three chemical barriers and made it into your urine. That's why in the urinalysis, if you see one of those, it's a really big problem. So now we have looked at the structure of our this is the renal corpuscle. This is the renal corpuscle. So the renal corpuscle is made up of two things. It's made up of a vascular component, which is represented by your glomerular capillaries. So these glomerular capillaries are blood vessels. So the first component is glomerular capillaries. The second component is what? Is the renal tubules. So within this renal corpuscle, you have two components. One glomerular capillary, red, going into the peritubular capillaries. All red. That is your vascular component. And here, yellow. This yellow is your tubular system. That has your proximal convoluted tubule, and then the loop of Henle, which is a descending loop of Henle and an ascending loop of Henle. And then you have your proximal convoluted tubule, and then you have your collecting duct. All of that is tubule. So the tubular component is trying to make fluid one, urine, and the vascular component is trying to make fluid two, blood. Both of them are fighting in a one place known as the renal corpuscle. So on the exam, when I ask you on the short answer, describe to me what's happening at the renal corpuscle. You say the renal corpuscle is made up of two components, a vascular component and a tubular component. The vascular component is the glomerular capillaries that are bringing in the blood and trying to get the blood back to the heart. And the tubular component is trying to make urine and send it to the bladder. Then we also discussed what are the forces that interplay between these two fluids. So we know that in the cap, we know we always have two forces. How many compartments are fighting, that shows you each. So we have two compartments. Each compartment has its own force. So what are the forces? Obviously, we have a hydrostatic pressure. So we have hydrostatic pressure in the blood, and that is represented by the glomerular hydrostatic pressure. And you also have the hydrostatic pressure of the tubular filtrate. So the hydrostatic pressure of the tubular filtrate. And you also have the osmotic, oncotic pressure of the glomerulus. And you also have the oncotic or osmotic pressure of your tubular filtrate. Now you must understand how they work. The hydrostatic pressure and the oncotic pressure at the same compartment will never be in the same direction. One is the hydrostatic pressure is pushing the blood out, but the oncotic pressure is telling the blood to come back. In the tubular compartment, the hydrostatic pressure is pushing the fluid back into the glomerular capillary, and the oncotic solute pressure is trying to bring all the solutes back into the urine. So don't think because they have the same letter denoted at the bottom, they are in the same direction. No, they are in opposite direction. So when the blood is coming in, when the blood is coming into my capillaries, who is pushing the blood to be filtered out? This space I'm making it so tight, but it's actually wider than this. It's much wider. So that's how you will see there's two compartments. Compartment that is in the core, the blood, and a compartment that makes it through the three layers. So this compartment here, this is my filtrate in orange. This is my filtrate. So now the first compartment, you have the hydrostatic pressure pushing the blood out to be filtrated. But the oncotic pressure of the glomeruli is saying to the blood, come back, let's return to the heart. You don't have to go to the bladder. So you see that they're in opposite directions. Direction of urine, designate that as the positive direction because we're trying to make urine. So the pressure of the glomeruli, the hydrostatic pressure of the glomeruli is pointing outward into the urine. But the oncotic solute pressure is trying to bring back all of the solutes and trying to stimulate the solutes to undergo osmosis and move from an area of a lower concentration gradient to an area of a higher concentration gradient to come back and return to the heart. Then in the tubular, in the filtrate component, you have hydrostatic pressure that is blocking, trying to fight the hydrostatic pressure of the glomeruli and push against it and trying to send the blood backward. And you have the oncotic pressure of your glomeruli trying to bring in all the filtrate down into the urine. So in a sense, the two that are working together are the hydrostatic pressure of the glomeruli and the oncotic pressure of the filtrate. They are working in the same direction to make urine, while the hydrostatic pressure of the filtrate and the oncotic pressure of the glomeruli are working together, and they are trying to send the blood back into the right heart. Is this point clear? Is it clear? Okay. Are we clear with this point? Okay. Then we talked about all of this. That's just filtration. That is just, so now we have filtered. So now we have created a filtrate right here. And the blood has now moved into the peritubular capillaries. The peritubular capillaries are now going to be dealing with with the remaining processes. Filtration, that's all about capillary number one. In our, remember we said the portal system. You go through two capillaries before you go back to the heart. So the first capillary, the renal corpuscle, our glomerular capillary, is responsible for the filtrate. Our peritubular capillary is now going to reabsorb. Is going to reabsorb solutes like sodium. Is going to reabsorb glucose, nutrients. Is going to reabsorb amino acids and electrolytes. And not only that, the peritubular capillaries are going to send toxic waste in the form of hydrogen proton. And that's what acid base balance is all about. How we regulate hydrogen protons to be sent from the peritubular capillaries into our kidney tubules. And the last step is water reabsorption. So water is going to be reabsorbed. So in the end, we're going to have a balance. The urine we're going to make, the urine that we need to make, this urine is either one of two. Is either a dilute urine or a concentrated urine. If it's a dilute urine, you know a lot of water is excreted and not reabsorbed, and a lot of solutes are being reabsorbed. So if the urine is dilute, that is because water is sent out in the urine and solutes are brought back into the blood. But if the urine is concentrated, then the opposite. That means a lot of water is reabsorbed and a lot of solutes are secreted. This is very important. So now your understanding of what makes a urine dilute, what makes a urine concentrated, will be accomplished when you think about what's happening in these processes. If my urine is dilute, that means there is a lot of reabsorption and there is a drop in the secretion. But if it's really concentrated, that means I'm absorbing a lot of water and I'm secreting a lot of solutes. So, and it depends from a person to a person. This is also regulated with hormones like ADH. What does ADH do? Antidiuretic hormone. ADH, always digging holes. So what does ADH do? ADH is all about reabsorbing water. So if ADH is secreted, what urine am I going to have? Am I going to have a diluted urine or am I going to have a concentrated urine? I'm going to have a very concentrated urine because what does ADH do? It reabsorbs water. It always digs holes to reabsorb water. And if I reabsorb water, my urine is going to be concentrated. So on this side, concentrated urine means plus ADH. ADH is secreted a lot. And on the other side, if there is dilute urine, that means minus ADH. That means I am not secreting antidiuretic hormone because from the name, what is diuresis? Diuresis means urination. So if I am secreting antidiuretic hormone, that means I'm trying to be anti-urination. So I'm trying to reabsorb the water. Dilute urine minus ADH, minus no diuresis. That's why the urine will be concentrated. But plus ADH, so ADH is anti-diuresis. So if I have ADH, that means I'm going to be anti-diuresis. I am against urination. So if there is plus ADH, that means my urine is going to be concentrated because I reabsorb the water. But if there is no ADH, then I'm going to have a dilute urine because all the water is going to leak out. Okay, that's a very important concept. Then the real high yield concept is what's happening here. You see, it's meant to be for a reason. Nobody erased this. This is really important because we will see this happening in the U-shaped vasa recta. This is happening in your ascending limb. This is happening in your descending limb and in your convoluted tubule. The ability of your kidney nephron tubule. This is happening at the tubular component, and the tubular component is communicating with the vascular component. These are red blood cells. These are red vessels. Remember, these are blood vessels. These are blood vessels. So now, if I want to reabsorb from the tubular component into the vascular component, I want to reabsorb glucose. So that is the lumen. That is where all my urine is. And that is the blood. If I want to bring if I want to bring glucose inwardly, glucose will be shuttled. It will be shuttled inwardly because it will hitch a hike with sodium. So remember we talked about how in the waterfall the water is flowing and they put a rotor with many blades and the water will strike the blades causing the rotor to spin, and then there is a magnet inside and a solenoid and a copper wire, and that will cause a magnetic field, and then they will take the magnetic field, convert it into electric field, and all that stuff. But what energy am I using here to spin this rotor that will bring glucose for me from the urine, the luminal apical side, into the basolateral side, and then into the blood vessels? It's the sodium. So I must always have a slippery slide for sodium to move in from the urine into inwardly into the epithelium of the tubules and then into the blood vessels. But that does not make sense because I know in my blood vessels I have a high concentration of sodium. Because remember we said, so in the when you guys did the action potential, you know that extracellularly sodium has the highest concentration. So how is it that although sodium has the highest concentration, I'm still able to have sodium flow from the urine side into an area that is high concentration? So how does that happen? Yes, very good. So the sodium potassium ATPase located where? At the luminal side or at the basolateral side? So Simon, think about it. What do you think? Where do you think the sodium potassium ATPase is? Do you think the sodium potassium ATPase is on this side, hydrolyzing ATP and then causing potassium to be sent inwardly and sodium to be sent outwardly? Or do you think it is found here at the luminal side where it will be hydrolyzing ATP to cause sodium to move from an area of a lower concentration gradient to an area of a higher concentration gradient? Do you think on potassium has to be in this direction? So what do you think is happening? Well, let's look at it in both cases. If the sodium potassium ATPase is happening in this direction, so now this channel is not going to work because the flow is in this direction. So now I am sending all the sodium into the urine. But if it's happening over here on the basolateral side, what's happening on the basolateral side is I'm sending all the sodium that's inside into the blood vessels against concentration gradient. So I'm making the core even more negative. I'm making it minus 70 millivolts. So now the sodium that's in the urine is going to find a slippery slide to go from urine into the epithelium lumen, urine into epithelium lumen to establish equilibrium. So they want a balance. They want to achieve an electrochemical balance. Electrobalance by charges. It is more negative here and they are positively charged. They want to make balance. Not only they want to make balance chemically or electrostatically, they want to make balance with diffusion. numbers. There is less number of sodium inside here and more number, so there is electrochemical gradient. So they will flow inwardly, and that will power the movement of glucose and amino acids and solutes. And once they get inside, there will never be balance, because the sodium potassium ATPase is always sending sodium out. So this guy says, I'm going in from the urine into the epithelium to establish a balance, an electrochemical balance. Once it gets in, it takes with it glucose, and once it makes its way inside the cell, sodium potassium ATPase will send it out. So there is always a net movement of sodium from the lumen into the epithelial cells of the tubules, and then outside through your sodium potassium ATPase. This gradient that is achieved by your sodium potassium ATPase is very helpful for shuttling the movement of amino acids into your blood. So this is happening here, and this is happening here. I'm reabsorbing all of my sugar, my glucose. I'm reabsorbing all of my amino acids. Not only it's this process is favorable for reabsorption, it's also good for secretion. So now I can have so this is a unit porter. It is carrying solutes and sodium in the same direction. But in secretion, what happens is I will use the same direction to move hydrogen proton in the opposite direction into the urine. Into the urine. Do you see now? So I use the secondary active transport that is generated by sodium potassium ATPase to reabsorb, but I also use it to secrete, to send out all of my hydrogen protons. And that is how I achieve my osmotic, that's how I achieve my acid-base balance. So my pH, my physiological pH, is 7.35 up to 7.45, up to 7.45. From 7.35 up to 7.45. If it goes below 7.35, I'm going to be secreting a lot of hydrogen protons into the urine. But if it goes above 7.45, I'm going to be reabsorbing a lot of hydrogen protons. Are we clear with this point? Okay. This point is golden. This diagram is very important. You guys understand it, and you're going to be able to reproduce it on the exam. Or does somebody need another explanation? Just remember, I use my sodium secondary transport to exploit the movement of glucose amino acids, reabsorption inwardly and in secretion to push out hydrogen protons. Okay, so let's do that. So this is my urine side, okay? And this is my blood side. What's happening is, just because I filtered urine, it does not mean it will exit immediately into the bladder. So if you take a sample from this urine filtrate that did not yet go through reabsorption and secretion, and then you take a sample of the urine in the bladder, they will never be the same. Why? Because the urine that has been formed as a filtrate did not yet go through reabsorption and secretion. So here, how do I achieve reabsorption? So in order to achieve reabsorption of glucose, to achieve reabsorption of amino acids and other solutes, I need a rotor. I need a channel here. And the channel will bring in inwardly from the urine side into the blood side my glucose, my amino acids, and my solutes. But in order for this rotor to start working, I need to power it with some form of energy. The energy I use to power it with is known as the secondary active transport. Why? Because it relies on a primary active transport known as the sodium potassium ATPase. The sodium potassium ATPase relies on ATP energy. It will hydrolyze ATP, and that hydrolysis of ATP will cause it to power the movement of sodium from the lumen of the tubule out into the. I think if you look, you will understand. Don't write so much. Just look. Try to understand first, then you can write. Yeah, that's going to be a lot better. So you know when you're looking in the board, it's like visual stimulation and auditory stimulation. But when you're just writing, you're just hearing things. So in this diagram, what's happening? That's the lumen of my urine. That's the urine leaving. That's here. That's my glomeruli. I'm done with the glomeruli. I filtered everything. These different forces are fighting each other, and now I have urine formed. Now, before this urine gets out, if you take a sample here of this urine that has just made it out, and a sample of the urine that successfully made it to the bladder, what will you see differently? You will see that the one that just made it out of the renal corpuscle is rich with a lot of glucose because there have not been glucose reabsorption yet. There haven't been electrolyte reabsorption yet. So you will see that there is a lot of solutes. So how did I successfully reabsorb them? So in order to reabsorb, I have rotors and I have channels here that are able to reabsorb from the urine amino acids, reabsorb from the urine a lot of glucose and solutes, but they rely on secondary active transport. And it is called secondary because it relies on a primary active transport, which is the sodium potassium ATPase. So the sodium potassium ATPase will hydrolyze ATP energy in order to move sodium out into the blood. So when sodium has been moved out against concentration gradient, that's why we use ATP, because it's not smooth. There's already a lot of sodium here. So in order to move it out, I'm going to need ATP. So all the sodium has moved out. And when all the sodium has moved out, now we will see that a lot of there is a net negative because sodium is positive. So there's a net negative millivoltage here, 70 minus 70 millivolts. And what's going to happen is that's going to stimulate the sodium in the urine. The sodium will say, wow, I'm positive. This is negative. I want to flow inwardly. I am more here. There is less sodium here. I want to move in chemically. So there is a double force, an electrostatic force and a chemical force. This is known as electrochemical concentration gradient. So it will flow inwardly. So the flow of sodium inwardly is going to be utilized by these rotors to reabsorb amino acids, glucose, solutes. Not only reabsorb, but it could also be used to send out hydrogen protons as sodium is moving in. So in all directions, sodium is always moving into the capillaries. But for hydrogen proton, it is sending hydrogen proton outwardly because we need to get rid of it, and it is bringing in solutes and bringing in glucose, bringing in amino acids because they are beneficial. Okay? Clear? Okay. Now let's go and see what happens in your vasa recta. And let's see what is so important about what's happening in your tubules. So now we have left our vascular component, and now we're dealing with a tubular component. So these are my tubules. Need yellow. Okay. Yellow will help you remember that this is dealing with urine. So the tubules are always yellow because they're always dealing with urine. So I have the proximal convoluted tubule. Then after that, I have the thick descending limb. Then I have the thin descending limb. Then I have the thin ascending limb. Then I have the thick ascending limb. Then the distal convoluted tubule, then I have two collecting ducts. One is in the cortex, on the top cortex, and one is at the medulla. This is my collecting duct. And now I'm going to send everything into the bladder. So what happens here? Well, the most important reason for so this is the tubule, and surrounding it is the interstitium of the medulla. So this is the kidney interstitium. So there is a lot of exchange. This is the vascular component around it, and this is the tubular component. And they constantly exchange. The solutes. So what I want to do ultimately is to concentrate the interstitium, to make the interstitium more concentrated. And when the interstitium is more concentrated, I'm going to have a dilute urine. And that is the normal urine. The normal urine for a healthy person is a dilute urine. So in order for me to have a dilute urine, that means I must be able to reabsorb all the solutes. And I must be able to send out water. So who's responsible for that? But the main goal is to make the interstitium really hyperosmolar, and make the urine hypoosmolar. And that is achieved by the reabsorption of the solutes and the excretion of hydrogen protons and water. It is your thick ascending limb who does all of that. Thick ascending limb. And the thick ascending limb is powered with a lot of sodium chloride reabsorbers. So what happens is they begin to create so this is the urine flowing inwardly. This is urine. This is the lumen, or the apical side. The thick ascending limb will begin to reabsorb a lot of sodium, this is into the blood. That's the what I drew before. So now we are absorbing from the urine a lot of sodium, and a lot of glucose is coming with it, and a lot of amino acids are coming with it, and a lot of solutes, and a lot of electrolytes like chloride. So all of these guys. So initially, at the glomerular capillary, when you look at this fluid, its osmolarity is very similar to the osmolarity of blood. How much is the osmolarity of blood? 300 milliosmoles. So 300 moles, 300 osmoles, 300 moles of these osmotic molecules. So if you look at before you enter into the loop of Henle, you take a sample from here, you see that the osmolarity of this filtrate is 300 milliosmoles. Once it gets here, the osmolarity, when you compare the osmolarity here, will it be 300? Will it be 300 like here? You know, you took a lot of chloride, a lot of solutes, and solutes have been taken out. So do you think it has more osmolarity than 300 or less? Yes. Less, yeah. Because I have removed. I used my channel to take out a lot of sodium, take out a lot of glucose, take out a lot of electrolytes. That will drop the osmolarity. Roman, are you following me? So when I remove with these channels, these rotors at the ascending limb, and I take two samples, this guy is 300, but this guy has went through a lot of reabsorption, so the osmolarity dropped to 100. to 100 milliosmoles. What do you think of the osmolarity in the middle? Don't forget, this is fluid too. That's the interstitium. That's the interstitium. At the beginning, the three of them are equal. This is 300. The one in the middle is 300. The one in the thick ascending limb is 300. But once these guys, these rotors, these protein channels start to reabsorb sodium into the interstitium, reabsorb chloride, reabsorb a lot of solutes and glucose, now there will be a disequilibrium where this is going to be 300. This is definitely not going to be 300. It's going to drop down to 100. And this guy used to be 300. What do you think going to happen to it? Excellent. It's going to go up. It's going to go up to 400. Why? Because I dumped into it chloride, sodium, and a lot of glucose and a lot of osmoles. So the osmolarity has increased. So now there is a disequilibrium. With this disequilibrium, a lot of water is going to flow now from both sides through bulk flow. A lot of water is going to flow into the interstitium. That's how I replenish my body. This is my body interstitium. So the water is going to be reabsorbed, and water is going to be reabsorbed, and then more urine is going to come in. So you're going to have round two. When round two comes, you're going to start all over again. This guy is going to here now it's 100 milliosmoles, and now he's going to the bladder. He's clear to be urinated. He's diluted. Now round two, more 300 is going to come in, and it's going to flow upward. It's going to get devoided. This is going to be 400. This is 100. He can go home. Water has been taken. So that's how it happens. This is really important. That urine comes in equal to interstitium equal to thick. But when it gets up to the thick, these thick ascending limb, these rotors will start to reabsorb all the solutes, making the interstitium the highest osmolarity. And that's very good because when the osmolarity is high, water moves from an area of a higher concentration gradient, like the thick ascending limb, into the interstitium. And also from here, it goes into the interstitium, and that drops my osmolarity to 100, and now we are in the bladder. And that's how it keeps happening. So you must understand how the shift happens. It is the thick ascending limb that throws into increases the osmolarity of the interstitium, and then water moves out, and then the last segment is now the osmolarity dropped, diluted, which is what we need. And then 100 milliosmoles. That's why the urine that comes out of the bladder usually is 100 milliosmoles. Then you have your cortical collecting ducts and your medullary collecting ducts, and that is where your ADH works. So antidiuretic hormone, if I want to take one, so this is the interstitium too, remember? If you're not yellow, you are interstitium. All of this is interstitium. Everything here is interstitium except inside the yellow tubules. So if I want to get water into the interstitium, I pluck all of these channels. So if they ask you on the exam, where does ADH work? It works here in these collecting ducts and in the distal convoluted tubule. It does not work in the ascending limb. It does not work in this. So here is the sodium potassium ATPase and these secondary active transporters. That's what's happening in your loop of Henle. But beyond the loop of Henle, the distal convoluted tubule, the collecting duct, then you will have your ADH working and plucking all of these aquaporins to bring in water from the lumen of the tubule basolaterally to the interstitium. Yes. Okay, in this diagram, so remember, the renal corpuscle is happening to give me filtration. So this is dealing with the whole phase of filtration. But when I move from filtration outward in the proximal convoluted tubule, then it's these guys. So now all of this fight here, oncotic G, hydrostatic G, oncotic tubular filtrate, hydrostatic tubular filtrate, oncotic tubular filtrate is just going to give me the filtrate that I will now have to project my filtrate into the proximal convoluted tubule, the loop of Henle, the descending loop of Henle, ascending loop of Henle, and then the distal convoluted tubule collecting duct. So these guys are happening in phase two. These are the ones responsible for reabsorption, because you see I'm reabsorbing a lot. They are also responsible for excretion of hydrogen protons. So there is a lot of channels here that hydrolyze the movement of sodium. Sodium is always moving into the interstitium. So sodium is moving into the interstitium, reabsorbing amino acids, glucoses, and solutes. But in the distal convoluted tubule, it's also moving into the interstitium, but in the opposite direction, sending out hydrogen proton. So this diagram here is dealing with phase 3, 4, 2, 3, and 4. Filtration, renal corpuscle. 2, reabsorption, concentrating the interstitium, making the urine dilute, and then also the secretion of hydrogen protons, and then the reabsorption of water with ADH, and then making a dilute urine. Clear? Okay. Any questions? Any confusion? Speak now or forever hold your silence. Are we all clear? So now let's go and see what's in the lecture slides. So as I said, on Tuesday, we're going to be beginning with a lab of acid base, the first activity of acid base, and then we're gonna do the first lab of digestive system, and we're gonna start digestive unit that week. So that week is going to be digestive and metabolism. And I don't think there's an experiment for metabolism, so it's just going to be a review day. But I'll send you guys an announcement this weekend to let you know exactly when the exam will be. What is this? Help. What do you mean call IT? I am IT. I don't know. Actually, Auntie helped me with IT, and I don't know that I should learn from that. Where is where is my? There we go. Oh, nice. It's inverted. Why does it look inverted in the camera? The camera is inverted. Okay, I'll see how it is. Okay, guys. So let's do fluid. Um. Okay, fluid, electrolyte, and acid-base balance. Okay. So the body water percentage varies. So a newborn's baby's body's weight is about 75% water. So the total body water content of an average young adult is 60% in men and 50% in women. So women have more concentration of what? Fats, lipid. Okay. So women's average slightly less because they have more adipose tissue, which displaces water. So the average total body water content of a 70 kilogram adult is 42 liters. So the obese and the elderly people as little as 45% by weight. So we are 60% water as men and 50% water females. Okay, fluid compartments. So areas are separated by selectively permeable membranes. And if you can follow, please go to the whole folder. Yeah, don't click on the concise one. Click on acid base fluid ZIP folder. This is the entire folder that was given to us by the publisher. So let's go through that. It will explain it. But you're not responsible for knowing all of it. Just responsible for the study guide that I will post up for you guys. If you do the study guide, solve it and turn it in before the exam, you're gonna do really well on the final on the exam. So, fluid compartments are areas separated by selectively permeable membranes and differing in their chemical composition. So, the major fluid compartment of the body is 65% intercellularly and 35% extracellularly. This 35% extracellularly, 25% of it is found in the vascular compartment, and this other 75% of that 35% is found in the extracellular fluid. In fact, there is a subtle space known as the transcellular fluid, known as your cerebrospinal fluid, the synovial fluid, the peritoneal, the pleural fluid, and the pericardial fluid, the vitreous fluid in your eyes, and the aqueous humor of the eye, and fluid of the digestive system, the urinary, and the reproductive tract. These are known as transcellular fluids. So I can ask you on a short answer. The extracellular fluid is divided into three compartments. The tissue compartment, the interstitial compartment we talked about, there is 8% blood plasma that is circulating in your blood vessels and lymph, and 2% transcellular fluid. Give me an example or three examples of transcellular fluid. You should write the cerebrospinal fluid, the synovial fluid, peritoneal fluid, the pleural fluid, pericardial fluid. These are so many examples to give from. So fluid is continually exchanged between the compartments. There's an intercellular and extracellular osmolarity are equal. So even though the compositions are different between your intercellular and extracellular compartment, the osmolarity is kept the same, 300 milliosmoles. Because water moves easily through the membranes, the osmotic gradient is never lost for long. If imbalance arises, osmosis restores balance within seconds. So osmosis from one fluid compartment to another is determined by the relative concentrations of solutes in each compartment. So most solute particles are electrolytes. So sodium salts in the extracellular fluid and potassium salts in the intercellular fluid. Electrolytes play the principal role in governing the body's water distribution and the total water content. So that shows you how we the movement of water between the major fluid compartments. So when you take a sip of water, water is reabsorbed, water is absorbed from your intestine into the bloodstream, into your circulatory system. And then there's an exchange between your circulatory system and the tissue compartment. And then part of it will be reabsorbed with your lymphatic system and returned back to the right heart. So water gain and loss. So fluid balance, when daily water gains and losses are equal, about 2,500 milliliters a day, the gains come from two sources. There is metabolic water that is produced as a byproduct of aerobic metabolism and dehydration synthesis. and there is preformed water that is ingested as part of the food that we take and the water that we drink. The routes of water loss are more varied. So how do we lose the water is varied. So we lose the water in urine, in feces, in expired breath, in sweat, and cutaneous transpiration. Is cutaneous transpiration the same thing as sweating? Okay, so water that diffuses through the epidermis and evaporates, but that is not sweating. Sweating comes from the sweat glands. So there's insensible water loss. Insensible water loss is not usually, you're not usually aware of it, and it is breath and cutaneous transpiration. So it just transpires and moves from one medium, your body, into the air, from an area of a higher pressure, your body, into an area of a lower pressure, the air. That's the only way for a fluid with the water or gas to move. So sensible water loss is noticeable output. So sensible water loss is the noticeable output, and urine and moderate sweating. So now you sense how much water that is being lost. And then there is obligatory water loss, and that is how much urine that must be excreted from your body. So for example, if they tell you you have to drink at least a liter and 200 milliliters of water a day, what does that mean? That means it is also the same amount that you must also excrete. Very good. So that is the obligatory water loss. So every day that passes by, you must not allow a day to pass by except that you have ingested a liter and 200 milliliters of water. Otherwise, you're going to make osmolarity stress on your kidneys. So, so how do we gain the water? We gain about 2,500 milliliters per day. 200 of those 2,500 milliliters are metabolic water. They are metabolic water. Okay. 200 of the 2,500 milliliters a day are metabolic water, and 700 milliliters a day is the water that's coming out from the food, and 1,600 milliliters of water that we must drink. I said 1,200, but 1,600 is as good. Losses. So if we take in 2,500 milliliters a day, we must get it out also. So the metabolic water that we take in, about 200 milliliters of water, is removed through feces. And the 700 milliliters of food that we take in, 300 of it is going to be removed and expired as expired air, and 400 are going to be transpired through the cutaneous layer. And the 1,600 milliliters of water that we drink every day, 1,500 milliliters of it are going to be excreted through urine, and your sweat is going to rid the body of 100 milliliters of that 1,600 milliliters that you have taken. So fluid intake is governed mainly by the sense of thirst. So the conditions that lead to thirst, you have dehydration, and dehydration reduces the blood volume and blood pressure, and it increases the blood osmolarity. So in dehydration, I have hypovolemia. So there is a reduction of the blood volume. And when I have hypovolemia, I have low cardiac output, I have low contractility, and I will have low heart rate. And I'm now heading in the direction of shock. So that drop, that drop in water in the body, how is it going to affect the osmolarity of the body? Because remember, the water was saving your interstitial fluid from hyperosmolarity, because water is a versatile solvent. So it dilutes the osmolarity. If you are dehydrated and you don't have the sufficient amount of water, then the osmolarity is going to increase and you'll have hyperosmolarity. You'll have osmotic stress. So there are osmoreceptors that respond to angiotensin 2 and rising osmolarity of the extracellular fluid. So these osmoreceptors are tasting. They're tasting how sour and how the osmolarity of your blood is increasing, and it will feed back and send a signal that causes the reabsorption of water through ADH. So ADH is released by the hypothalamus and it promotes water conservation at the distal convoluted tubules and stimulates the cortex to provide the sense of thirst, to direct you to go and replenish water intake. Salivation is also inhibited because it consumes a lot of water. So salivation consumes a lot of water, and we want to conserve water loss because we are dehydrated and we are under osmotic stress. We will decrease salivation. That's why patients who have dry mouth means that they don't have salivation. It means that they are really dehydrated because the body is trying to conserve as much water as possible, and that impairs the formation of saliva because saliva is made up of water. And that's how one of the major signs and symptoms of dehydration and low cardiac output and hypovolemia is the dryness of the mouth. The long-term inhibition of thirst. So the absorption of water from the small intestine reduces the osmolarity of the blood because we said water is a versatile solvent. And the long-term inhibition of thirst stops the osmoreceptor response and it promotes the capillary filtration and makes saliva more abundant and more watery. So when we look at dehydrations, thirst and rehydration, if dehydration takes place, that would lead to two effects. The first effect is increased blood osmolarity, and the other effect is reduced blood volume. So the first, these both of them are, they constitute for me a metabolic stress. So one stress, dehydration, will be translated in your interstitial compartment as two separate stresses. The first one is the increased blood osmolarity, and the second one is reduced blood pressure. And each one will be solved by a separate system. So let's look at the system that will repair the increased blood osmolarity. The first system will lead to, firstly, a thirst response, where there will be a stimulation of the hypothalamic osmoreceptors. So the hypothalamic osmoreceptors, they will sense that there is hyperosmolarity. They will cause a reduction in salivation, and they will cause dry mouth, and they will cause a sense of thirst. The other stressful stimuli, which is the reduced blood pressure, that reduced blood pressure will be sensed in the kidneys, and the kidneys will cause the secretion of renin, and renin will be converted by angiotensin into angiotensin 2. So we will have angiotensin converting enzyme, and it will convert for us angiotensin 1 into angiotensin 2. Angiotensin 2 that was converted from angiotensin 1 in your lungs will cause a stimulation of the hypothalamic osmoreceptors that will lead to dry mouth and a sensation of thirst. That sensation of thirst will lead you to go and search for water and drink water. Once the water, you have intaken the water, that will cause coolness and a drop in the hyperthermia, the overall hyperthermia in the body. That will cause cooling and moistening of the mouth, and that will cause a rehydration of the blood, and a distension of the stomach and the intestines. So the only way to control the water output significantly is through variation in the urine volume. So the kidneys cannot replace water or electrolytes. They can only slow the rate of water and electrolyte reabsorption and secretion. And changes in the urine output linked to adjustments in the sodium reabsorption, and water will flow in the movement of sodium because water is a versatile solvent. Then antidiuretic hormone will provide means of controlling water output independently of sodium. And as osmolarity of the blood rises, ADH is released and the kidneys will reabsorb more water and produce less urine. This opposes the decline in blood volume and a rise in the osmolarity. As osmolarity of the blood drops, your antidiuretic hormone secretion will be dropped and the kidneys will reabsorb less water and produce more urine. And this will lower the total body water and compensates for hypertension. Okay. This slide is very important because it shows us that we begin with a dehydration stressful stimuli, and dehydration is represented by a drop in water and an increase in sodium concentration. That will cause an elevated blood osmolarity, and this elevated blood osmolarity will cause a stimulation of the hypothalamic osmoreceptors. These stimulated hypothalamic osmoreceptors that were stimulated by a drop in water concentration and an increase in osmolarity. So, what does dehydration represent? If I tell you to describe for me, what is dehydration? Is it just the loss of water or a drop in the amount in the overall homeostatic water range in the body? What, in addition to that, also represents dehydration? It happens, it also happens concomitantly. So it's like a seesaw. When water drops, what else also increases in concentration? Solute, osmolarity will increase because water dilutes the osmolarity. So dehydration is not just the mere loss of water, it is the loss of water and the increase in your osmolarity. Yes? So hypovolemia means that I have a loss in water in my blood compartment. But dehydration is a more generalized term. So I have lost blood from my interstitial compartment, I have lost blood from my intravascular compartment, and I have lost blood intercellularly. So dehydration is more dangerous. So if I drink the water and I uptake the water from my intestine, that water will not only be stored in the extracellular compartment, it will have to nourish my intercellular compartment and also the intravascular compartments. I have three compartments in two. I have an intracellular compartment and an extracellular compartment, but the extracellular compartment is divided into two. In fact, it is divided into three, not two. The extracellular compartment is divided into intravascular compartment. It is divided into interstitial compartment, and also, what is the third compartment? Transcellular compartment. Very good, which represents cerebrospinal fluid and represents your vitreous humor. How about we do a lab? You guys seem so excited for doing a lab. Yes. You just had your hand up like that? Okay, no problem. Okay, do we dare do a lab? Let's do a lab real quickly. Let's start. You guys seem a little bit bored of lecturing. How about let's do exercise 10? Okay. We go back to fun. Let's just do one. Let's just do one lab. It's so little. It's just like a one. Yes. Okay. Um work with somebody. Work with Munir. Work with Munir, and remember I told you last week, see me after class so we can sort through what's happening with your physio works. Okay. Okay. Okay, that's fine. So work with somebody for now, and then we will fix that. Okay, so let's do hyperventilation. So in hyperventilation, our objective is to introduce pH homeostasis in the body and is to understand the normal ranges for pH and the partial pressure of carbon dioxide, to recognize the respiratory alkalosis and its causes, and to interpret the oscilloscope tracing for hyperventilation and compare it with a tracing for normal breathing. I remembered a couple of things actually regarding acid base analysis. Okay, yeah, exercise one. It's not exercise one. Hyperventilation. The first, we're always doing the first activity. Okay, so when we're talking about acid base balance, there's a very important concept that you guys need to keep in mind. The first concept is that we obviously have the pH as one of the homeostatic variables. That's why we keep it and we ensure that pH remains within the range of 7.35, maximally up to 7.45. Anything below or above that will represent a sort of metabolic or a sort of pH stress. So, in order to survive, my healthy green range is going to be 7.35 up to 7.45. Above that, you have a little yellow survival range, just a little yellow. Okay, and then you're gonna go into red. Red is death. Once you hit red, it's death. So healthy range is 7.45 up to 7 point, maximally 7.48. Above that, death. And here 7.33. Below that is going to be, you're going to be fighting for your life. Every loss of 1 point represents logarithmically represents death. So it represents millions parts. It represents parts per million. Represents so much logarithmic increase in the hydrogen proton concentration. So in order for me, so this is my safe green range. Below that, below 7.35, that's going to be acidosis. Above 7.45, that's going to be alkalosis. So, very interesting. I must remain in the green range at all times. And this variable, this pH variable, represents for me my acid-base balance. So who's responsible for ensuring my acid-base balance? We talked in the past that our respiratory system, our respiratory system is a very important regulator of acid-base balance. We know that carbon dioxide, when it enters our body, it will increase hydrogen proton concentration, and that will represent a stress. And to exit and throw out and send away carbon dioxide, that will drop hydrogen proton concentration. So I know my respiratory system is a very important regulator of my acid base balance. Who else? So that is 33% of the regulation. Who else is responsible for my who else is responsible for the second 33% of acid base regulation? That is my renal system. We talked about how in the renal system you have the powering movement of secondary active transport, and we reabsorb sodium into the interstitium, and we have that rotor that secretes hydrogen protons, and that will represent for me how the renal system is responsible for ensuring that I have the proper physiological range of my the proper physiological range for my hydrogen proton. So if I am at acidosis, that means I have high hydrogen proton concentration. What happens is I will hyperventilate and send out a lot of hydrogen, a lot of carbon dioxide, and that will ultimately drop my hydrogen proton concentration, and that will increase my pH into the healthy green range, and then I will send a sensory input through an afferent neuron that will be integrated, and that will end that reflex, that hyperventilation reflex, homeostatic reflex. Also in my kidneys, if I have acidosis, that means my pH is low, hydrogen proton is high, I will increase the secretion of my hydrogen protons from the interstitium into the tubules, into the renal tubules. What is the third? What is the third system that regulates Fermi? What is the third system that regulates for me my acid base balance besides my respiratory system and besides my renal system? And if you say the answer, you have to give example. So just guesses like the nervous system, the digestive system. No, you have to say how. What is the third system responsible? Is it actually a system, or is it just a group of things that work as a system? Such as, yeah, what's the answer? Excellent. So the buffering system, the bicarbonate, excellent, 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 excellent. The bicarbonate